Hello, my name is Bob Phillips. I'm one of the children's doctors here at the Leeds Children's Hospital. I'm also a researcher at the University of York. I was funded by the NIHR, that's the National Institutes of Health Research, to do some studies looking at how we can better improve the management, that is, improve how we look after children and young people who have febrile neutropenia, which is a complication of cancer chemotherapy. So febrile neutropenia is a situation where when you're being treated for cancer, you often have chemotherapy which is immunosuppressing, that is, it reduces your immune system. One of the things that opens up is the possibility that you will get an extremely severe infection, and that infection will come on very, very quickly. And sometimes these infections are fatal. And so what we say is that if you get a temperature or you feel unwell in a sort of infectious way, you have to come to a hospital, and then you have to get brought into the hospital and given extremely powerful, what we call broad spectrum antibiotics, just in case it's one of these severe bacterial infections. Now what we know is that for every 5 kids that turn up, only one of them will have one of these severe infections, and the other 4 will have something else, a virus, some inflammation may be triggered by the treatment or by the cancer or just by something else, and didn't really need the antibiotics. But at the point they come through the door, you can't see the difference immediately between those that definitely need it and definitely don't. And so we give everybody these powerful antibiotics. One of the problems with that is that it means that the more antibiotics we use in the world, more chance there are that the bacteria will become resistant. That is, they won't be killed off with the antibiotics. And so a lot of what we're doing in the world at the minute is about trying to reduce antimicrobial resistance to keep the antibiotics working. Lots of different ways have been tried to try and work out the difference. And one thing that people had suggested in the past was that maybe we could do some monitoring of inflammatory proteins in the blood. Now, as you probably know, the blood carries lots and lots of things. It carries white cells that fight infection, red cells that give you sort of the energy stuff, platelets that stop you bleeding and bruising. And it's also got lots of proteins in it that send messages to other bits of the body. Now, some of these proteins are released when our body is attacked by something. So that might be a bacteria, might be viruses, might be a fungal infection, or it might be that we've fallen over, or a cancer is making things inflamed, or we've had an operation or something like that. But we know that different ones get released in different ways. And what we needed to do was to work out, was there a marker in the blood that would tell us who was the seriously ill one versus who were the ones that weren't seriously ill, potentially as they walked through the door. We collected together all of the information that had been done in previous studies, and it was suggestive that these 3 markers that we've tested might make a difference. One was called interleukin 8. Now this is a neutrophil attracting protein. Neutrophils are the cells that come and eat bacteria. And when they go up, it's like, bring in the neutrophils, come and eat the bacteria, come and make things better. One of them was called interleukin 6. We know that this gets released when pieces of bacteria, particularly certain types of bacteria, land on cells in the body, and it like triggers an explosion of this protein to be released. The protein itself can be measured, and it also makes a secondary protein come out called procalcitonin. Now we measured IL-8, IL-6 and procalcitonin. Why, you might well sensibly ask, did you measure procalcitonin if all that is a product of the IL-6? Now that's a really straightforward thing. In order to measure IL-6, you basically need to do a proper chemistry style experiment with a real lab technician who knows exactly how to dribble things in, to measure stuff. It's really quite complicated, and it can take a good day to get a result back. Now, the procalcitonin, they have these massive machines that do loads of measurements. Stick the blood in one end, within an hour, the result comes out the other end. So although it's connected it's practically an awful lot easier. So if that is the one that makes a difference, or is as nearly as good as the one that takes a scientist a day to do, that's a much more practically useful test to be using. So NIHR funded this study to say, does it make a difference? Can we draw things together? And we took a study that took place over the course of about a year, and we had over 200 different families say that they would take part. Now, these were saying they would take part when they were having chemotherapy. Not everybody has these fever episodes. 60 patients then did develop an episode of fever, and about 30 of them came into the hospital when we were doing the tests and when we had the ability to do that. So we've got data on 31 patients who had a variety of things that were going wrong with them at the time. We collected all sorts of information. We collected how high their heart rate was, what their temperature was, what their blood count was, whether they looked poorly or not by the doctor looking at them, these 3 fancy tests, some boring tests that we did at the same time, and we followed them all up to see, what did we think was the cause of that infection in the first place, how long did we give them antibiotics for, what else did they get going on, all that sort of stuff. And what we found was those patients that looked really quite unwell to the doctors had massively high levels of these inflammatory markers, the procalcitonin, the IL-8 and the IL-6. Now, this is great, but it was in the patients that looked unwell. So the question arises, does the blood test tell you anything over and above what you would have seen anyway, just by having a trained doctor in front of you? And the way that I think about this is, you know what a duck looks like, and you know what a yak looks like, well, these big cow-like creatures. You can tell the difference by taking a blood test and sending it to a lab to do a full genetic analysis. And in about a week's time, the lab will send back and will say, that blood came from a duck, that blood came from a yak. It's a lot easier just to look at the thing in the first place. The more we looked at the data, the more it tells us that actually all the blood test does on that first day, it tells you what you already know. Sick kids have high levels of this. It doesn't really separate out as they walk through the door the ones who are going to be sick and the ones who aren't going to be sick, apart from what you can already see. But there was something else in there. The ones whose consistently low levels of these inflammatory markers, we kept treating them with antibiotics because they kept having temperatures. And they really didn't need those antibiotics because it never went on to get poorly. They never went on to have a bacterial thing that needed the antibiotics in the first place. And so that led us on to do another study. So not can you tell the difference when you walk through the door, because we know that that doesn't add anything over and above what we've got in front of us, but can we use this instead to stop the antibiotics earlier? Because normally we'd wait until your temperature had stopped. Now, if we can stop the antibiotics earlier, that means you can get home earlier, and it means we can use less antibiotic. So we moved on to do a very simple study to look, could we do that? Could we reduce the antibiotics by looking at the trend of this procalcitonin? And if it stayed low, even if they had a temperature, cutting back on the antibiotics. And what this one has shown us is that we can do it. It might make a difference. Only really saves an average of half a day, which sounds like nothing. But that's an average. What it means is that for 3 out of 10 patients, they get home 2 days earlier than they would have done. The other 7 out of 10 doesn't make a difference. It doesn't make it any longer, but it doesn't make it any shorter for them. Half a day doesn't sound like a lot, but for those 3 patients who get home a couple of days earlier, that's pretty good. And actually, if you add up all of those half days of the hundreds and hundreds and hundreds of episodes, you're saving quite a lot of antibiotic. And so our research is going on to see, can we make a difference that way? What we showed was that as you walked through the door, measuring these tests really didn't tell you much more than we knew from just seeing you and seeing whether you look poorly or not. But tracking them forwards in time might well still help us in cutting back on antibiotics. And so, whilst this study sort of didn't help us, it did, because it said, don't do that, and gave us another avenue to explore instead. So the next level now, we've got our this might work. We've got data where we pulled it together from other things, like in grown-ups in intensive care and older people with really bad lungs. We know that this can be used in other situations to reduce antibiotic load. We know it can be used to reduce antibiotic load in the tiniest babies, the premature neonates. And now we've got a tiny bit of proof that it might work in children's oncology, in young people's oncology. And so now what we need to do is go back out there to different funders to get another trial up and running where we test our standard way of doing things against our new brilliant idea and see, does it really make a difference? Does it make enough of a difference that it's worth putting the hassle into extra blood tests, the cost into extra blood tests, and can we make that difference to the patients in getting a chunk of them home earlier and antimicrobial resistance in reducing the amount of antibiotics that we need to give over that time?